Thanks for joining back. In this lesson, I'll be talking about proteins, the second biological molecule that we'll be covering. Proteins are integral to almost every cellular function, structure, and are involved in the macro scale in the regulation of the body's tissues and organs. Proteins are made up of subunits called amino acids. Amino acids. Though amino acids have their own roles in the body, such as serving as neurotransmitters in the brain, they are the building blocks of proteins. Amino acids, if you remember from last lesson, I was talking about the side groups that are found commonly in biological molecules. And amino acids have them as well. So you have an amine, let's use a different color. You have an amine group, and you have a carboxyl group. And if you remember, in carbohydrates we had a carbonyl group, and this is different. This is a carboxyl group, and I'll show you exactly where these are, but I'd like you to take a minute to look at the structure and think for yourself where these two side chains might be. While you do that, and while you're looking at the structure, I'll be telling you about the elements that make up amino acids. So amino acids are made up of carbon, hydrogen, oxygen, nitrogen, and sometimes it's made up of sulfur. And if you noticed, the same elements seem to repeat in these biological molecules. And this is because all of these molecules are made up of combinations of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. And we'll be talking about this when we go over nucleic acids. So, if you remember, carbohydrates were made simply of the CHO, and in proteins it also includes sulfur and nitrogen. So now that you've taken a minute to look over this amino acid structure, I'm going to be going over these where the amine and carboxyl groups are. And if you haven't had the time to do that, just take a second and pause the video, and when you come back, I'll go over it. So if you're just joining or if you're waiting, our amine group is here with the nitrogen bonded to 2 hydrogen atoms. Our carboxyl group is found on this side, and you can see that it's double bonded to an oxygen and then single bonded to an oxygen and a hydrogen. Now the last part and the most confusing part, or at least right now, is this R. What is this R? It's variable. So it allows us to identify all of the different amino acids that I'll be covering. And so this original structure will be common while this R group will be changing for all of the amino acids I'll be going over. And there are 3 main groups or subgroups of amino acids. You have essential amino acids, non-essential amino acids, and conditional amino acids. In humans, there are 9 essential amino acids that cannot be produced by the body and instead are taken up in food. And so now I will be going over what these are, but also the amino acid structures and functions or properties. And this will relate to the functions of these amino acids. So, unlike carbohydrates, in proteins, the monomers, these amino acids, are incredibly important. And you need to know all of the names, structures, and properties of all of these amino acids. So I'm going to zoom out for a second, because I'm going to be writing over this. And you can screenshot or take a picture of all of these structures, and then I'll be going further and diving deeper into what these mean. Okay, cool. So the first amino acids I'll be talking about are the charged amino acids, and these are positively charged. Now, I mentioned that there were 9 essential amino acids. 3 of them are arginine, histidine, and lysine. And again, this doesn't mean that other amino acids aren't essential. It's just that these cannot be produced by the body. Now, if you look at these structures as well, you can also tell that there is the amine group at the bottom and the carboxyl groups on the sides. And this is different from the way I drew it initially, and this is just so we can highlight exactly what these subgroups are. In addition, similar to carbohydrates, I won't be drawing exactly the carbon or the hydrogens, and so when you see these areas with the ridges, you can assume that there's a carbon here, and this is commonly drawn. If you want to see exactly where these carbons are, you can look up images and you can see a more defined structure. Now, you also have the two negatively charged amino acids, and that's aspartic acid and glutamic acid. For polar amino acids, you have serine, threonine, asparagine, and glutamine. So threonine is a essential amino acid, and serine, threonine, asparagine, and glutamine, what it means by polar is just that you have a negatively charged and a positively charged end. And so it's not fully charged, but it has this two poles in its structure. Charged and polar amino acids have the property of being hydrophilic. And hydrophilic means that it loves water. It loves water. Yeah. Hydrophilic means that it loves water, which means that it's attracted to water. And so when you're looking at the structures of proteins, these will be found on the outside of the protein, and it will be shielding the nonpolar or hydrophobic, phobic meaning fear of, so hydrophobic amino acids. Phobic. But before I go into hydrophobic amino acids, I'll be talking about these special amino acids. So in cysteine, you have this sulfur, and that creates disulfide bridges in the tertiary structures of these amino acids. I'll be talking about this further in a second, but we'll go on to selenocysteine, and if you look at these two, you can see that they have similar structures, but that the sulfur is replaced by a selenium. And this isn't commonly found in proteins, but which is why I've marked this with a star. In addition, there's 20 common amino acids, and this is like the 21st amino acid. And so the other ones are more commonly found in protein structures. Next, you have glycine, and what's special about glycine is that it has it's the simplest structure, and so it would only have this hydrogen as its R group. For proline, as you can look at it, you can tell that the NH2 is not fully shown, and this is because the side group is attached to your amine group, and so that's kind of what's special about this amino acid. Next, the polar amino acids. Of these, the essential polar amino acids are valine, isoleucine, leucine, phenylalanine, and tryptophan. And so here we have all of our 21 amino acids, and we have our 9 essential amino acids. So those are arginine, histidine, lysine, threonine, valine, isoleucine, leucine, phenylalanine, and tryptophan. Cool. Now I'll explain how amino acids come together to create proteins in a later lesson on transcription and translation, but an important characteristic of proteins is protein folding. And this is extremely important because it dictates the function of the proteins. So protein folding. The shape of proteins, again, is extremely important because it dictates the function. And misfolded proteins can cause diseases such as Alzheimer's disease because it doesn't do its function. And so in the example of Alzheimer's disease, you have a buildup of these misfolded proteins because the body is trying to do a function, but it isn't able to complete it. So in these structures, you have classes of structures that make up this protein folding process. You have a primary structure, which then becomes a secondary structure after it is changed a little. Then you have a tertiary structure. And finally, you have a quaternary structure. So your primary structure is extremely simple. It's just a sequence of amino acids. Your secondary structure, on the other hand, is local interactions between stretches of your polypeptide chain, and it includes alpha helix and beta pleated sheets. So if for an example, if you have, let's say, 6 amino acids, and these amino acids are charged and these ones are not, or I guess we're going with gold for nonpolar, so gold. Because these ones are hydrophilic, you'll be having the charged or polar amino acids on the outside and the hydrophobic amino acids on the inside. And that's just an example. But in a larger scale, you will have alpha helix structures. And so these are just spirals. And you have beta pleated sheets. And so these are just common structures that are found or created due to the secondary interactions. So this is beta pleated sheets, and this is alpha helixes. In your tertiary structure, this is the overall three-dimensional folding that is driven largely by interactions between these R groups. An example of that would be your disulfide bridges. So let's say you have a hydrogen and a sulfur, and you have a sulfur and a hydrogen. You have these disulfide bridges that are created, and this will link other parts of your protein together. And so post your tertiary structure, I know this seems like you have you finished folding this amino acid because you finished the secondary interactions between stretches of the polypeptide chain, and then you actually have your side chains interact already in this tertiary structure. But your quaternary structure is multiple of these proteins interacting with each other, or polypeptide chains interacting with each other to create your final protein. And so your final protein is made up of multiple of these polypeptide chains that are edited throughout your primary, secondary, and tertiary structure. So now that I've gone over protein folding, the last thing that I'll be talking about is the types and functions of different proteins. And I'll just be glossing over this, and we'll be talking about specific proteins and their functions in later chapters. But some examples are enzymes, which you've probably heard of before, and these catalyze reactions that occur in cells. You have structural proteins, and these provide structural support, as in the name, but also helps the shape. And so an example of this would be collagen, which actually makes up your nails. Is this fact? I don't think it is. Collagen, nails. Which an example of this is An example of this is collagen, which actually makes up your nails, skin, and cartilage. Another function of proteins is transport. So you have transport proteins that move molecules across membranes and from one place to another in the body. An example of that would be hemoglobin in the blood, and it moves around oxygen to other parts of the body and allows cellular respiration to occur. Next, you also have motor proteins. And so these convert your chemical energy into mechanical energy, and so that's found in the flagella and the cilia of different cells and bacteria. And so an example of that is myosin, which is found in muscle fibers. This is a next you have storage proteins. And storage proteins store amino acids and metal ions. You have signal proteins, which send messages to specific locations in the body and the cells. You also have receptor proteins, which can be found in the cell membranes of cells. And so these receive messages. And finally, you have gene regulatory proteins. And so these regulate gene expression and are found in the nucleus. And we'll be talking more about these when we go over gene expression, but also during transcription and translation. Cool. So we've gone over the majority of proteins, and we'll be going over a bit more in later chapters. But just as a summary, we have amino acids, which are made up of an amine and a carboxyl side group with an R group, which is variable. And this decides what amino acid you're looking at. And you have 21 amino acids, and 9 of them are essential for humans. You have charged, polar, special, and nonpolar amino acids, and it's incredibly important you remember the properties and the structures that are linked to these names because they dictate what occurs during protein folding. And protein folding dictates the function of your protein, and it's separated into primary, secondary, tertiary, and quaternary phases, with primary just being the amino acid chain, secondary being the interactions between the polypeptide chains within the polypeptide, tertiary being the interactions between the actual side groups, and finally quaternary being between multiple polypeptides and finally creating your final protein. Your final protein has different types or functions, and so you have enzymes, structural protein, transport protein, motor protein, storage protein, signal protein, receptor proteins, and gene expression proteins. And so this wraps up our lesson about proteins, and I'll be joining you next time to talk about nucleic acids.